I'm glad that you made it back for part three. This is a continuation on a lab that was written so that it gives you a pretty good view what's going on in a clinical lab. Hopefully, by the end of this, you feel comfortable. So, we've already talked about differential and selective media. I'm going to talk about it again. And so, uh, we have different types of media that we use in the diagnostic lab. We can use sheep blood auger, which is a differential, is not selective. Chocolate auger, which is really the way we used to call it, it was burnt blood auger, but you heat it a little bit more and it turns into this darker chocolate auger. And there's some microorganisms, some sexually transmitted disease organisms that will prefer chocolate. And my favorite of all times, Mcconkey auger. It's a selective media and a differential media built into one. It's one of my favorite plates for looking at gram negatives. So other types of isolation media is liquid. We can use the thioglycolate broth, which is really what we look for is the differences on oxygen utilization. So ones that can utilize oxygen will grow higher up at the top. You can see the denser growth areas. Some that can't handle the oxygen, you'll see it trying to grow away. This one's all the way down. This one's at the top and at the bottom. And so it tells you a little bit about the types of if they are anaerobic or not, and it's a really nice test to do. Another simple test is the Simmons citrate agar test, and this is to find media to determine if an organism can use citrate as a sole carbon source. And this is some can, some can't, and it's often used to differentiate between the gram negatives, the Enterobacteriaceae, in organisms capable of utilizing the citrate as a carbon source. The enzyme citrase, any word that ends in ASC is an enzyme, hydrolyzes citrate into oxoacetic acid, which is an acid, and will change the pH. So the green is what it starts off as, and then turns blue, and that's the alkaline pH indicator. A positive test, the blue on the right, is citrate positive. Klebsiella pneumoniae and Proteus mirabilis are examples of these, and it'll turn those pretty to the blue. Mannitol salt auger is taking advantage of osmotic pressure. Its concentration of salt is 7.5%. So that means there's 7.5 grams of salt in every 100 mLs of the media. And that's a lot. Members of the staff can tolerate it, and other genera may try to grow, but typically they don't grow so well. Here's the Serratia marcescens over here. You don't see any growth, and here you see Staph aureus growing. And not only that, but it is breaking down the sugar mannitol, and you can see that because the acid is diffusing from the breakdown, and the acid will turn it yellow. And again, it makes a nice it's a differential type plate and selective, so it's both. And again, you can see the mannitol breakdown and forming fermentation products. But you can see this is a single colony isolates streak, so we know it's not contaminated. And you notice the pH area, there's not enough microorganisms there to really change that pH, but the preponderance of it is obviously, and the same here. You can see that they both grow, but this one is breaking down mannitol, which makes it a great differential. So this has got Staph aureus and Staph epidermidis, and it doesn't break down the mannitol, and it's a nice differential for that. So it's a great, simple, easy, cheap test to run. And so the yellow is positive for mannitol fermentation. This is negative for mannitol fermentation. Actually, yeah, so you can see there's no yellow on that, but they both grew. Here's another positive test. You can see it streaked out in a small area. Depends on how long the plate's been incubated to see how much. Then again, you can see here's Staphylococcus aureus, Staphylococcus epidermidis, and then E. coli streaked on this plate. E. coli can't handle the truth. No, it can't handle the salt concentration, so it won't grow. But the staph will grow just fine, and it breaks down mannitol. So here's a really great a collection of information that we can use in our court of law to convict. If our pathogen happens to be a Staph aureus, this would be a mannitol positive, and it is growing on the high concentration of salt. So it just gives us some nice touch points there to add. Here's my favorite, my Mcconkey Auger, and what we have is lactose fermenting colonies. You can see the colonies actually turn to pink. Now, if it's E. coli, it actually kind of leaves a sort of a I don't know, a halo effect around the colonies. But this is one of the few tests that it doesn't really change the media; it changes the colony color. Non-lactose fermenting colonies don't colorize, and they just stay kind of bland and boring. So Proteus vulgaris, Salmonella typhimurium kind of fall into that, and Staph aureus is really having a hard time with the Mcconkey Auger because it uses the same dye as we used in the gram stain, and because it is for gram positives, it clogs the pores of that, and the that dye will keep the organism from growing. So the Mcconkey Auger contains the crystal violet dye, and it adds that sort of selective characteristic to it. Here, E. coli has that unique, it does ferment, and it leaves sort of this halo effect, where Enterobacter aerogenes also ferments, but it doesn't have that halo. It's sort of unique, uniform throughout. So you can see we can use this auger very nicely for selective purposes. So again, this is something, the colony is lactose positive, and it ferments the salts around here, so it salts out, and it's so characteristic of Mcconkey, and it's usually a good indicator of E. coli. Another lactose positive, but it's growing. And here's a lactose negative. It's growing, but it's clearly not breaking down lactose. But again, it's one of the few medias that actually has the colony changing color, which is really nice. Here's a mucoid. This is Klebsiella pneumoniae, and this is Citrobacter fruendi. And they have characteristics. This one tends to clear around a little bit, but it's also lactose fermenter. This is non, but you can see this mucus. It looks like somebody blew their nose on the plate. So here's another Mcconkey Auger showing you dark red, and it's taking up two dyes for this particular organism. Has something in the membrane that's binding it, but it's still looks like it's doing a fair job of fermenting, but something else is going on with it. I always like to show some of the weird stuff that goes on from time to time. Some kind of mobile genetic element or something, I'm not sure. Brilliant green auger is another one. Sometimes we use it, sometimes we don't. In a lab that sees a lot of salmonella, we typically use a brilliant green, and it's E. coli on plate B. You can see E. coli has been cultivated. The color of the plate has changed to yellowish to yellow, so it's a pretty good indicator. If you look at C, a salmonella species cultivated on plate C, note that the color of the plate has changed from yellowish to dark red, so it's a really nice indicator. Again, it's usually used, salmonella is a very close cousin to E. coli, and these two are really hard to distinguish. Sometimes even under a microscope they can appear to be very similar, but this using brilliant green auger will definitely nail the difference between the two. So we use differential media to help decipher these types of organisms on various things, and this auger that I was just talking about, brilliant green, does a fair job of that. Eosin methylene blue auger gives you, we typically call it the EMB plate. It's selective differential used to isolate fecal types of bacteria, coliforms. ESNY and methylene blue are pH indicator dyes that combine to form a dark purple precipitate, which is sort of a greenish sheen. It's really cool on a plate. I like to do it just for the cool factor. Sucrose and lactose serves as a the fermentable carbohydrate, and you can see these vigorous fermenters very nicely on these. Normally, if you have lactose negative like Proteus vulgaris or Salmonella or Staph, you don't see that. But look at the green sheen we see with E. coli. Again, it's a nice diagnostic test. Enterobacter orogenies always does that. It seems to be peripheral on is sensitive to the air or concentration of some of the chemicals in the media, and the edges that tend to turn it to a dark black. But the rest of the lactose negatives pretty atypical. Again, here's another ESN why I like to use it, and we're looking at the lactose positive. You can see it's most likely E. coli we're talking about. There's a lactose positive, but it doesn't produce the sheen. The metallic green does tell us it's utilizing both the sugars, and then the lactose negative one there as well. All right, let's see, ESN. Okay, let's go back here. A differential media only. So blood auger is only differential. And the principle of blood auger, of course, is looking for some factor that breaks down blood. And we call this hemolytic type of reactions. And it's unique to some pathogens, and we take advantage of it. So usually what we do to make blood auger is that we make usually 1 liter, 2 liters at a time, and we add these types of things that you see, and then sheep blood. If you're lucky, you can get it defibrinated. So in other words, it's not clotted and all that. And you add the red blood cells, and it's usually what they're adding about 5 grams. 50 mLs in 100 mLs, so in 1,000 mLs. So it's not a whole lot, but the thing is the media has to be cool, and you need to cool it down to about 45 to 50 degrees Celsius, and then you can add the blood, usually pipetting it in, and then you mix it very carefully so you don't get bubbles and things, and then you can pour it on the plates. And when you do, they get these nice red plates, and when we streak microorganisms on them, they'll grow. And the thing is, it's nice is if they produce a hemolysin, we can see it, beta, alpha, and then gamma. It says none, but gamma is typically we see with nothing. So the betas are bad. Typically, these are your Staph aureus. They tend to be Pseudomonas aeruginosa, Streptococcus pneumoniae. Here's your Klebsiella, which is gamma, which is negative. So here's the hemolycins again. The greenish is the alpha, and you got to be really, really careful when you look for these as to age of the plates, because usually what causes this is hydrogen's peroxide production, and it will turn, it sort of rusts the red blood cells because it has iron, it turns it green. But you look for the reaction right after incubation, you can see the greenish areas, then you know it's an alpha. Beta is clear, it's that golden color or clear totally, and so you can see alpha, beta, and gamma. And I'll leave, of course, the information will be on your slides. But the reason we do this, look at the difference between a strep and a staph, and in terms of the hemolysis that we see. And again, the hemolysis that we see with the strep and the staph are uniquely different. Let me show you again. So A, B, C. C is obviously, beta is clear all the way through. B is also a beta, very clear. And then A is also a beta, and you can see the clearing. Now it does have that greenish tint, but if you were to examine the plate, it would be clearly beta. alpha, beta, oops, and uh, uh, alpha, beta, and the beta is here. This is greenish, so that is alpha, and then C is gamma. So this is clearing, goldish color, and that is what? I heard not all at once now. This is beta, bad. B is what? Alpha, right? Because it's somewhat green. And then C, there's nothing going on, so it's um, what? Gamma. So just to review, here's the breakdown of that. Now, it's easy to get flustered on that, but don't let that happen to you. It is just sit down and you want to look and think about the beta being the worst, beta bad. That's why I say it that way, so you'll remember that. So beta, then alpha, and then gamma. So that should work for you. Differential media tubes, slightly different. We use the media in a liquid form, and in the liquid they will turn the color. Sometimes we can even determine gas and things like that. We have different types of media. We've already talked about this: plates, slants, broth, and then deeps we use for various things. But we still even get the patterns on the slants, even though it's media that was heated and the tubes were slanted, and so that they give you this huge lawn area because they were poured hot on a slant, and then you let them cool and they'll harden with a slanted auger on that. So slant's very useful, and we draw on the surface of the of that auger using our sterile loop with the bacteria, and of course they have various names for those. I'm not going to hold you to it, just know that they are. If you remember, when we are working with tubes, that we use the cap and let that be held by our pinky finger. And again, just the flaming technique is you want to make sure that you flame the neck of the tube, get your sample, and then flame it again, and then replace the top. And you do that for each of the tubes. Anytime you go in, you got to flame the top, so it creates a heated area movement outward of the tube, and that keeps things from going inside. And that's a technique that we use. The objectives of a type of tube array that we use is the triple sugar iron types of tubes, and you can see representations of ones with different types of microorganisms. Here is a control, and then you can see various other tubes with all sorts of things going on. This is usually the very cheap, low cost, of course, but it gives us a lot of data, and the data is useful in court to identify these microorganisms. And so we utilize the information here, and I'm going to help you sort through this. It's really easy once you see the tops. If I were to draw a magic line right across here, this is the tops and the bottom, where they call it the butt. So the top and then the butt of the tube. So this is yellow, yellow, red, yellow, red, black, and there's a reason why it turns black, because hydrogen sulfide is produced and it turns the media this jet black of sulfur, and it's a nice indicator. But the top is red and the butt is black. Yellow at the top, black at the bottom, red and black, and then the top is red, and here is the butt is yellow. So we have all these different determinations, but look, we can identify a series of microorganisms using a test like this, the gram negatives. And again, this is for the gram negatives, and of course it's part of the Enterobacteriaceae group, and we look for as many of the pathogenic traits as we can, depending on if we can identify it. But if we can't, we just use a simple triple sugar iron auger test to help identify it. So I have here, so you can read through the slides, the write-up of what's actually going on. So I've kind of highlighted in black some of the key parts. Differential basis, so, the top yellow and the red and all that, it's based on fermentation of three different sugars: glucose, lactose, and sucrose. Now, what's interesting is that you have 10 parts of lactose, 10 parts of sucrose to only 1 part of glucose. And there's a reason for that. It's really clever. And then a peptone to have it break down a protein. So phenol red and ferrous sulfate are the indicators for the pH indicators for acidification, and then the ferrous sulfate is for the hydrogen sulfide production. That's what's responsible for turning it black. The acid base indicator, phenol red, is incorporated for detecting the carbohydrate fermentation, and that's indicated by the color change. It's usually the orange red to yellow, and it makes it really nice. In the case of oxidation, decarboxylation of peptone, alkaline products are built and the pH rises. The change of the color medium from orange to red to deep red gives you that additional handle. Sodium thiosulfate and ferrous ammonium sulfate present in the media detects the production of hydrogen sulfide and turns that black color in the butt of the tube. It's always in the butt though. So glucose is utilized first by fermented organisms, and then the entire media becomes yellow, but remains acidic. 18. See, we have to use kind of a timing issue like we've seen in other types of media. So reading it at either 8 to 12 or 18 to 24 hours is really important to determine the types of fermentation that we see. But it's a poor man's way of looking and identifying and getting the names of these microorganisms. So in addition to glucose, lactose, and sucrose are fermented, a large amount of fermentation products formed on the slants neutralizes alkaline amines and renders the slant yellow. And that's if it's read in the 18 to 24 hours. So then you'll get the formation. So if you read them early on, you can get the red and yellow, and then the hydrogen sulfide comes on a little bit later from 18 to 24. And sometimes it produces gas. You can see the media just lifting up, and so we can say that reported as gas fermenting. So here is all the variations that we can have. Now, for whatever reason, the yellow or where we have the formation of acid, we refer to that as A. So the top is A and the bottom is A. If there's some bubbling or movement of the chunking of the auger, we call it A. This is red, yellow at the top, red, yellow at the bottom, and a little bit of gas is formed. We call it AA gas. We see usually at the butt. It's not so nice as that drawing, but anyhow, it's A, and you want to read the tubes early. I come in after 12 hours to look at the tubes, and then the black gets darker and darker, so we know that the butt was yellow at one time, and then it starts to get consumed by the hydrogen sulfide. If there's gas, we can report it as AA with gas, with hydrogen sulfide present. AK, so the reddish where we have the media not being the indicator not showing the presence of an acid at the top, so we see the yellow and then red. So the top is yellow, and we refer to the red on these tubes as K, so it's an AK, and this tube is a KA. So the top of the tube is red and the bottom is acid A, and with gas, if it chunks in, you see that it's a K, red at the top, A, acid at the bottom, and Ka, and then we see the hydrogen sulfide. If it's chunking or having gas, you'll see Ka with gas, and then there's Kk where it's red at the top and bottom. So all of these help identify the characteristics, and one of the challenges I have for you, I'm not going to ask you this on an exam, but looking at Ka, AA, looking at some of the differences, look at all the information that we can obtain, and remember, we can use this information to identify these microorganisms later on. So that was what this process was about, to identify microorganisms, and a lot of this data will be provided for you to help identify your unknowns, and so you want to be able to interpret the tests and then identify the microorganism, and from that, now you won't obviously perform the technique to create these, but that data will be given to you for your clinical identification of the unknowns. And so it's interpreting these to figure out what your unknown is, and you'll have a chart to go by, and so that's what you have. So that's what we have for now, and we'll come back later when we talk more. See you then.